Without further ado, it's my honor to introduce Ivan Corbeski. So Ivan studied biochemistry at Goethe University in Frankfurt, Germany, and then he did his master's studies in Deutsch Group on enzyme dynamics. And afterwards, with a short interlude between his master and PhD, he moved to field of structural biology in BVOT Center for Biomolecular Research of the Utrecht University in Netherlands. I'm sorry if I pronounced the first part wrong. So there he worked with as a Marie Curie fellow in the with Rolf Bohlen and Hugo van Ingen on NMR spectroscopy, and then he obtained his PhD in 2019. And his PhD was focused on the structural and functional characterization of a histone chaperone involved in DNA repair, which is also the topic of his talk today. And then since 2020, he first started his postdoc in Bernec Lab in the Institute of Science and Technology in Austria. And then since 2021, he moved to Lab of Cavendish Group in the Department of Biochemistry, University of Zurich, Switzerland. So welcome, Ivan. The floor is yours. We are listening to you now.
Okay. Yeah, thank you very much, Judith, for the kind introduction, and the fragile nucleosome team for the opportunity. And I welcome all the community members from around the world. And I want to start my talk by saying chaperone. Just because this is my favorite word in science, but I also think it has a very interesting origin from social life. And the term actually comes from the Latin capa, later turned into the French chaperon, which means hat. And this hat was worn generally by people who looked for good behavior in society, and later the people themselves who wore these hats became the chaperones. And so by definition, a chaperone is actually a person who accompanies and looks after another person or a group of people. And to illustrate this for our everyday life, a chaperone is simply an adult who, for example, takes care of children, for example, when they want to go to the amusement park, because in the absence of a chaperone, kids want to do all crazy kinds of things, like ride a roller coaster upside down, which is of course dangerous. So the chaperone makes sure that they get properly and safely onto the roller coaster of their lives. Now, after this short excursion to the origin of the term chaperone, we can go together to the molecular chaperones that I'm interested in and I want to talk about today. And for that, we just have to shrink this roller coaster and put it into a cell nucleus. And we arrive at our genome in the form of chromatin with the nucleosome as the basic unit. And the children of chromatin are the histones, which form dimers and tetramers through the so-called handshake motif. And the general model says that one H3H4 histone tetramer binds first to DNA to form a tetrazome, which then binds to two H2H3 dimers to form first a hexasome, and in the end, the nucleosome with the histone octamer wrapped by the DNA. And the chaperones are histone chaperones, of which there is an entire army which control histone folding, transport, and most crucially, and what I want to focus on today, the nucleosome assembly and disassembly mechanism. The question is, how do you do it? What are the interaction steps and intermediates involved? And there are different models for nucleosome assembly, and one very well understood model of nucleosome assembly is that of in vitro salt dialysis, which starts at 2 molar salt and at 4 degrees, because the histone octamer is only stable under these conditions, otherwise it dissociates. And the ions of the high salt concentration simply stabilize the histone octamer. They neutralize the positive charges of the histones to prevent their repulsion, and they drive their hydrophobic interactions to stabilize the histone octamer and prevent unspecific interactions with DNA, which can then, as the salt is dialyzed out, start to interact with H4, which has the highest highest affinity DNA binding site. And then as the salt is dialyzed further, the DNA can wrap around the histone octamer to form the nucleosome, which is stable under physiological salt and temperature conditions. Now, for nucleosome assembly mediated by histone chaperones, there are two different models. And the first one is a thermodynamic model. In the absence of a histone chaperone here to the left, if you just add histones and DNA together, they unspecifically interact with each other, leading to irreversible aggregates and making nucleosome assembly simply impossible. But the archetypical chaperone NAP1 can stabilize the histones and reduce their energy so that nucleosome assembly becomes the more favorable path. In the alternative, the kinetic model, it is actually that the binding of a histone chaperone reduces the energy for the transition state in such a way that that nucleosome assembly becomes the kinetically favorable reaction so that the nucleosome is formed. But the question here is, how might such an activated transition state look like? And what are the steps then towards nucleosome assembly? And the chromatin process that we want to elucidate this in is in DNA repair, in particular non-homologous end joining, which is the emergency pathway to repair DNA double-strand breaks. So in this, when there is a DNA damage, chromatin has to be decompacted. The DNA repair machinery recognizes the break site, repairs it, and then chromatin is compacted again, which is in agreement with the excess repair restore model of DNA repair. The question is, what happens with the histones during this process? And a central factor in the repair machinery is the protein APLF, which holds the machinery together, and it has histone chaperone function in its acidic C-terminal domain. So we set out to study this C-terminal acidic domain of APLF. And so here you see the domain architecture of APLF at its N-terminus, and in the central region it has binding sites for the other DNA repair factors. At its C-terminus, it has a poly-ADP ribose binding zinc fingers to bind PAR, which is a signal for DNA damage. And at the very C-terminus, its acidic domain with histone chaperone function. And this is highly negatively charged, and it has a histone binding domain with several aromatic residues in it. And at the very C-terminus, it has a Kr motif, which is rich in lysines and arginates. And so we looked at by NMR spectroscopy at the interaction of APLF with the histones. And what came out of this is that APLF acidic domain is intrinsically disordered, and it has two different binding sites for two different types of histone complexes. A binding site for H3H4 containing two aromatic residues which function as anchors to stabilize the complex formation, a double anchor 1 for binding H3H4 and a double anchor 2 with two other aromatic anchors to stabilize the interaction with H2A, H2B. And we showed by ITC that when mutating these anchors to alanines, this either largely decreases histone binding or it completely prevents histone binding. So these aromatic anchors are really crucial for histone binding. But also, since there are these two different binding sites, this already suggests that APLF could potentially bind both histone complexes at the same time. And we tested this by size exclusion chromatography under conditions under which the histone octamer is not stable. So that's why when you just have a histone octamer mix under low salt conditions at a room temperature, the two different histone complexes are separated. But when you add APLF to this, you get one stable complex. And by native mass spec, we could actually show that this complex consists of the histone tetramer, H3H4, and two copies of the H3H2B dimer and two APLF. And by ITC, under similar conditions, we showed that indeed two APLF bind the histone octamer with a very high affinity and also with enthalpy of binding that is more than double than if you just sum up the enthalpies of the individual histone complexes. And this is because of synergistic binding of APLF to the histone octamer and the induction of additional histone-histone contacts. And finally, after this, we tried to reconstitute the complex, and we managed to determine the crystal structure in which part of the DNA of the nucleosome is effectively replaced by two copies of APLF to form what we call the chaperonosome or a chaperone body. And the APLF is here fully disordered, and it nicely traces the positively charged surface of the histone octamer, and with its aromatic anchors, it binds into hydrophobic pockets on the histone surface, except for this tyrosine, which is in a flexible linker between the two different binding sites. And these binding sites are at the H3 alpha 1 alpha 2 patch and the H2B alpha 1 alpha 2 patch. And they are additionally aided by hydrogen bonds and salt bridges. And the function of these interactions between APLF and the histones is to interfere with DNA binding over both DNA gyres. And APLF causes both a steric and an electrostatic clash with the DNA. Now, in our crystal structure, we have just a part of the APLF acidic domain, but with the help of cross-linking mass spec data, we could also model the rest of the APLF acidic domain in two different conformations. In one, which is a proximal conformation in which this very C-terminal part binds to the same H2H2B as the anchors, or in an alternative conformation to the opposite H2H2B. In any case, in both of these conformations, to largely interfere with DNA binding, and both protomers of APLF form this cradle in which the histone octamer is embedded and covering most of the histone octamer DNA binding interaction sites, except for the small region around the diet. And to test these interactions and their functional relevance in vivo, we did some cellular assays. And in DNA damage tracks, we could see reduced recruitment of APLF when the 4 aromatic anchors were mutated, actually a similar reduction as if the entire acidic domain was deleted. And in pull-down assays, mutations in either the H3H4 or the H2H2B binding sites lead to reduced binding of both histone complexes. And this indicates that in the cells, the substrate for APLF might indeed be the histone octamer. We then tested in vitro if APLF, what APLF could do to the histone octamer. And by using a short DNA fragment, we could actually show that APLF can form a ternary complex with histone octamer and with DNA, while when you use a bigger DNA fragment that is sufficient to form the nucleosome, APLF is actually excluded from this complex. And then we tested if APLF indeed can form nucleosomes with an MNase digestion assay. And what we could see is that as you increase the concentration of APLF, and we quantified this, you get more nucleosomes. Now, to determine the effect of the aromatic anchors on nucleosome assembly efficiency, we did in vitro chaperone assay that is based on native page and DNA detection. So when you take DNA and add an excess of histones to it without a chaperone, this leads to aggregates that are removed from the assay, leading to no signal. But when the chaperone is added, aggregation is prevented and nucleosomes are formed. When you mutate the aromatic anchors, there are less contacts between the chaperone and the histones, leading to more unspecific interactions between the histones and DNA. Still some nucleosome assembly efficiency, but much less, and more aggregation. And when all four anchors are gone, the chaperone cannot control the histones, which can again fully aggregate with DNA. But very interestingly, actually crucially, as determined by ITC, these mutants bind the histone octamer with very high affinity, actually similar affinity as the wild type. And this comes due to enthalpy-entropy compensation, which is a strongly pronounced phenomenon for interactions involving intrinsically disordered proteins like APLF. And this leads to little changes in binding free energy. And with these data taken together, actually means that the conformations of the mutants, which allow for more unspecific interactions between the histones and DNA, must also be possible in the wild type APLF. Only there, the aromatic anchors can interfere with DNA binding and lead to a more controlled interaction between the histones and the DNA. And this translates to a nucleosome assembly mechanism under physiological conditions in which APLF in the form of the chaperonosome stabilizes the histone octamer through its negative charges, and it leads the histone octamer to start interacting through the highest affinity binding site on H4, so that then the DNA can wrap around the histone octamer to displace APLF, which stays bound to the histones until the nucleosome is fully formed. And looking at the energetics, so while NEP1, for example, can fully explain the thermodynamic model of nucleosome assembly, with APLF and the chaperonosome structure, we now think that we can explain how a kinetic model of nucleosome assembly could look like, because here the chaperonosome is the activated state of the histone octamer, in which the histones are already pre-assembled in the form of the octamer, ready to be deposited onto DNA to form in a stepwise manner the nucleosome. And how can you remember this mechanism? You just have to remember the roller coaster. The chaperone hugs the children and then puts them one after the other and onto the roller coaster of life. And now that I'm at the end of my talk, I would like to thank my chaperones and the people that came together to make this story. And as I told you, the histones from dimers and tetramers through a so-called handshake motif. And as I showed you, whereas they're divided alone, the chaperone brings them together. And in the same way, we did the same thing. We put our hands together to bring this story to life. And the people that contributed to this are my supervisor, Hugo van Ingen, in the NMR group in Utrecht, together with my promoter, Rolf, and our NMR spectroscopy, Hans. We did the crystal structure in the lab of Tisha Sixma at the NKI, together with Xiao Hu. At the Hubert Institute with Francesca Mattiroli and Bruna, we did the chaperone assays. Cellular studies were done at LUMC in the group of Hypophonaticum, together with Kees and Wouter. The mass spec we did in the lab of Professor Albert Heck, together with Domenico. At the EMBL in Hamburg, we did some SAC studies in the group of Dimitri Svergun, together with Melissa. I did ITC at the University of Leiden in the group of Marcellus Übing, together with Anlus. And I also want to thank my current supervisor, who is still accepting me to give talks about my previous work, Amedeo. And of course, we got funding from the Dutch science organizations and internationally from Marie Curie, INEXT, and Instruct. And with this, I want to thank you for your attention, and I'm happy to answer questions.
So thank you, Ivan, for your great talk. So we already have 2 questions in Q&A box. And everyone asked, so very interesting model. I understand that APLF primarily binds to H2A, H2B, but your crystal structure shows APLF having contacts with H3, H4 as well. Sorry. Do you think APLF differentiates between H2A, H2B dimers or H3, H4 tetramers as binding partners? Does APLF has increased affinity towards any one over the other? Does the FHA domain or the KU binding motif have any contribution toward the chaperone activity or the DNA binding ability of APLF?
Yes, yeah, thanks. Yeah, very good questions. So APLF is able to bind H to H to B individually, H3, H4 individually. So with H3H4 under conditions when H3H4 is a tetramer to APLF binds to each H3H4 dimer, so then to the tetramer, and in the case of the octamer, so APLF can, as the structure shows, bind basically the histone octamer configuration, in which the two different histone complexes form the octamer. And as for the affinity, so yeah, so from ITC has a high affinity for the histone octamer. Yeah, 150 nanomolar is what we measured, but I also have to say that that was at 300 millimolar salt. So potentially at lower salts, so more physiological, let's say 150, I would expect that affinity will be much higher. The reason why we could not work at such low concentrations is because of the methods that we used. ITC requires a lot of protein, and at very low salt you would get aggregates under those conditions. So we had to go a bit higher with the salt. As for the chaperone activity, so the FHA domain, so actually APLF is very nicely integrated into the non-homologous end joining machinery with the FHA domain binding to the peptide of XRCC4. Then it has APLF has a Ku binding motif with which it binds to Ku, and then it's connected by a long linker to the PBZ domains of APLF, which can bind poly-ADP ribose, which is a modification on DNA and also on histones. And the PBZ domains are very close to the acidic domain, so I can very well imagine that in DNA repair, the binding of poly-ADP ribose can assist in the chaperone function of APLF. But this certainly needs to be tested.
Okay, thank you for answering this long question. I mean, Jacob has raised hand, but I will ask. I would like to ask him to write his question because we are out of time. And there's also 3 more questions. So even it would be nice if you could answer them in the chat. So now I will give the floor to Matteo so we can continue with Maria's talk. Thank you, everyone.